and regenerate their cells and now consume them that can. And our job, actually, is to help them share their data and visualize it in context.
Yeah, you go test those. Thank you. Okay. Christian, I'm sorry. Just mute other.
You can mute them yourself, okay, but you will have to manage it the whole time.
Thank you, Terry. Good morning.
Say something like, hey, you're not speaking.
And
that feedback noise.
Thank you to Nick for the invitation to speak this morning. So first question, how many people here have heard of ICTA?
Should I mute?
Okay. Now, how many people here who aren't getting direct salary benefits from ICTA have heard of ICTA? Okay. So, good. This is an opportunity. As Terry said, ICTA was launched on December the 4th last year, and it's version 2 of the previous Clinical Translation Science Institute.
Example that it's going to be a moving target.
This course really facilitates, but it also teach and mentor investing how to do community research, especially that for people that that's critically important for the success of
Okay. Can you hear me now? Can people hear? Okay. So this is Ajay Pillai. You are listening to the webinar on the cutting-edge informatics tools for illuminating the druggable genome, RFA RM 1800011. It would be good if people mute themselves, and if you want to ask us questions while I'm talking, please use the chat feature, or else we will open the microphone at the end for questions. So with that, I'm going to restate some of the things that I said while I was on mute. So this webinar is being recorded, and this will be made available on the IDG Common Fund website, and I'll point out the URL during the presentation. Okay, so first thing I want to point out is that this is Common Fund RFA, Funding Opportunity Announcement. And I wanted to point out that the Common Fund actually has certain very special characteristics. These include that the programs in the Common Fund have to be transformative, have to be catalytic, short-term, and goal-driven. They are synergistic and enabling and add value to multiple NIH institutes and centers, and they are cross-cutting. They're cross-cutting in nature, and they provide novel solutions to specific challenges. And so they're not like standard R01 projects. And specifically, I'll talk about Illuminating the Druggable Genome, which is the program under which Aegis, this RFA, is being put out. The goals of the program are to improve our understanding of the properties and functions of proteins that are currently not well studied within commonly drug-targeted protein families. So there is a division in effort between the experimental parts and the computational parts within the IDG program. The experimental part focus primarily on 3 drug classes of families, protein families, GPCRs, ion channels, and protein kinases. The informatics activities are in general proteome-wide, and the IDG program is also interested in demonstrating the feasibility of a systematic approach to looking at understudied targets, and the approaches adopted are potentially generalizable to other protein target families. So I will, because you'll be joining an existing program, it's important for you to know what the structure of the program is, what it looks like right now, so that your applications can reflect characteristics of the program that are important. So the IDG network as it exists today is made up of experimental groups that are in sort of yellow creamish color here. These are the experimental centers that generate data and reagents. We have an outreach function that is specifically separated out in the Resource Dissemination and Outreach Center. We currently have a Knowledge Management Center, which is purely informatics. That's the top part of this blue circle. And informatics tools, this is what is represented by this RFA. This thing shows, the picture shows the interactions with external data sources, as well as our existing and interactions we hope to develop with the outside community of academic investigators, biopharmaceutical partners, foundations, and international organizations. You will see that there are a lot of URLs in this talk, and there are good reasons why they're there. I'll briefly describe what these URLs do. So the first URL in here is about where the webinars are. I can see. And this is where the Common Fund 1, the first URL is where the recording is going to be made available. In general, if you want to email about the funding opportunity, you should use the help email. The Druggable Genome Consortium URL is next. An important subpage on that consortium page describes the scientific strategies and decision trees that are going to be used by the experimental centers. They also describe the types of experiments that that are undertaken within the IDG program right now. So these are important background material that would be useful for your application. So a few brief things to say about the experimental groups. There are 3 groups of experimental centers that have been established within the IDG. One that looks at GPCRs, another one that looks at protein kinases, and a third one that looks at ion channels. The PIs and co-PIs are listed here. This is in a previous phase of Illuminating the Druggable Genome project. One of the outcomes was creating a small molecule which was an allosteric ligand for 2 dark GPCRs. These compounds have been made available within Sigma, Sigma for purchase, and this is an example of the kind of resources that the IDG hopes to put and make available to the community. A little bit about what the Resource Dissemination and Outreach Center does. So their primary aim is to package up the resources that are being generated, especially the experimental resources, as well as the data sets that are being generated within the IDG program, make them FAIR compatible, and make it available to the community through existing resources that are not funded by the IDG program. They will also be made available through the IDG program portal as well as the IDG website. They also take part in making sure that the community can utilize these resources and do other outreach activities. The Knowledge Management Center, its primary role is to computationally collate and make available a range of information that's scattered around in different resources that either the NIH or the community supports in various ways. The KMC has established pipelines to bring together data about all proteins that we know about, as well as information about diseases that they are involved in and any chemical reagents or biological reagents that are available for these targets. And that data is collected in the Target Central Resource Database, and it is made available to the community through the Pharos website. The Pharos website has multiple features that enables faceted searching. It provides detailed views of individual proteins, and it has a robust REST API that is accessible. More URLs, and I'll take you through what these are. The first one is the base URL for Pharos. The next one takes you through a description of the APIs that are available that you can access through Pharos. Then there's a general help page. The data sources that are collected within the TCRD and Pharos is described in the next two locations in the data sources. The Pharos has made available through its fact page various how-tos in using Pharos, how the existing visualizations within Pharos can be used is a separate section in the fact page. Finally, probably most pertinent to this RFA is this section called Pharos and IDG, and that describes some of the underlying technologies that are being used to build Pharos and to collect the data within the TCRD. You can download the entire data set that's available in Pharos as MySQL dumps. There are web tutorials that are available, and finally the overall program website. Okay, now I'm going to get to some of the specific parts of this RFA and this funding opportunity announcement. So the program contact for this RFA is Jean-Claude Zenclusen. Remember, this is a Common Fund program, not an NCI project, and I work at the NHGRI, so we work in all disease areas and tissues, and that's of interest to the NIH. So it just so happens that NCI is managing this particular RFA. That's why both the program and the grants contact is at the NCI, but you essentially work with a project team that spans different institutes at the NIH. Just basically take you through some key dates. The application opens up for earliest submission roughly a month from now. Oh, on June 9th, not a month from now, 10 days from now, I guess. The letter of intent is due on June 9th, and I think this is something that helps our colleagues in CSR to organize a good panel for review. So I understand that it's not essential for you to write, it's not required that you submit an LOI, but it helps us create an efficient way to get to do the scientific merit review. The application due date is July 9th, and as usual, it's end of business at your local time from wherever you are. The scientific merit review will take place in October, November 2018, goes for a second review to the Advisory Council in January, and the earliest start date would be April. And typically, these are computer systems that may go blink at the last date, so it's probably a good idea if you try to submit the application earlier. Some more administrative details generally about touching on things that help investigators who are more familiar with the R01 format. In any decisions that we make, the resource sharing plan will be considered carefully. It will also be considered carefully by the review group. In general, NIH applications now require that you provide an authentication plan for key biological assets that you would be using. In general, NIH also wants you to consider sex as a biological variable when your applications. The research plan, there's a 12-page limit. The data sharing plan does not count towards this limit, and as usual, do not use the appendix to circumvent the page limit. As I said before, U24 grants are not standard R01 review criteria, so it's good that you read the RFA and you read what review criteria are going to be used. Typically, in addition to the existing ones, there are some very specific things for this RFA that are mentioned in the review criteria section. I will briefly touch on them towards the end. As usual, applications deemed to have highest scientific and technical merit will be discussed and assigned an overall impact score. There are no appeals to the initial review that will be accepted. And the recommended applications will receive a second review by NCI advisory councils. And my colleague Mark Caprara from CSR would be happy to address any questions regarding peer review that you may have today or later on. So I do want to spend a few minutes, or at least one slide, talking about the resource sharing plan. So the primary goal, and with the example that I indicated with the GPCR ligands, the primary goal of the IDG program is to make data and resources readily available to the broad scientific community. So it's important in the resource sharing plan for the awardees to manage data, resources, protocols, tools, and software in a manner that would achieve this goal. So, open source, for example, would be highly useful and make life easier in evaluating these things for everyone. So all applications, as I said, must include the resource sharing plan, and applicants should indicate in the application their willingness to abide by data deposition, quality control metrics, standardization, metadata requirements, data and software release, and public copyright licensing policies developed by the entire consortium. And finally, any tools that you develop through this effort have to be made available within Pharos. And in the Pharos FAQ, we have provided, I think, reasonable amount of detail on what you would need, what sort of underlying technologies are being used. And we are happy to take additional questions, and if you have any on that part. So this last URL highlights the software sharing section of the RFA, just to keep it handy for you. In terms of budgets, the total amount of funds available for this FOA is $2 million per annum. So these are not direct costs for individual applications. This is the entire budget that's available. If you are new to doing NIH budgets, I've provided a link which takes you through gory details of how to do this. Specific individual application budgets are limited to $300,000 in total direct costs. As usual, budgets need to reflect the proposed project and the work involved. The maximum project period for this work is 2 years. So specifically, I want to talk about what we want to achieve now through this FOA. There are two major classes of things that I think the IDG program would be interested in. One is building predictive models, and the second one is building new visualization tools. So predictive models have to focus on improved ways of associating understudied proteins to biological function or disease or pharmaceutical relevance. These models, if you propose to build such predictive models, you have to do a small component that would validate them experimentally, that your models make sense, that they are reasonable, the predictions that come out have some validity. These are not deep experimental validations, so use the total amount of funding available judiciously to allocate to what you think would be within your system a reasonable experimental validation. And as I repeated before, these models have to be made available to the community using Pharos. This is a technically difficult thing because you have to keep this up to date and current. If you're using data sets that keep changing in the background, that get updated, then you have to figure out how these models would get updated, and you have to think about automating some of these processes. The second kind of applications that would be responsive would be to build new visualizations. So as you know, there is a lot of data and information that's available that's scattered throughout various resources on the web and also within Pharos. Pharos supports certain types of visualizations. We do want to figure out ways in which the community can actually prioritize targets, understudied targets that they want to study by looking at as much of the evidence available about them and their relevance to either diseases or a biological condition or pharmaceutical experiments, pharmaceutical situations. So new visualizations that can be launched from within Pharos would be very valuable for getting the information out to the community. As I promised, I wanted to bring to your attention this specific RFA-related review criteria. So in the significant section, reviewers are going to assess whether it is likely that your approach is going to make accurate predictions about the biological roles of understudied proteins. So they are going to assess that the proposed experimental validation approach is doable, is suitable, and is appropriate for generating preliminary experimental validation. In the approach section are the data sets that you bring to the table to either build predictive models or to add to the Pharos collection that you propose to visualize. Are they appropriate and justified to the aims that would enhance the aims of the IDG effort? Are non-IDG resources that you are proposing to use, are they available freely, or do there exist constraints on their use? And as I hinted at before, you have to think in your software development efforts, things like containerization, providing provenance, and fairness metrics. And finally, are there plans to make these tools available within Pharos and to the community adequate and appropriate? With that, I come to the end of my presentation. And I think we are happy to take questions. I'm going to try and unmute everyone. Is that what we're doing? Okay, you can unmute yourself if you have a question. If you want to ask a question on the chat, please feel free. That would be a way to anonymize yourself. You can use the email. Okay, so I will handle some of the questions that we have seen up till now. So the first one is, what percentage of the overall budget is expected to be devoted to bench validation? Well, there is no unique answer to this question. It depends on the situation. So I think in general, the experimental validations are not the primary part of the primary mission of this FOA. You should think about all the other software engineering and computational tasks that you propose to do first. There are criteria that require you to create robust software, which is more unusual than your standard computational approaches. So those things are going to take some funds, they're going to take some resources. Experimental validations are important because, if you're making a prediction, you don't want to just put up a prediction without some way to see that, people should believe the predictions that come out. So that's my general and specific answer, but I'm happy to take more discussion later on this in more specific details if you want. The next question we have is this FOA limited to the 4 classes of drug targets outlined in the, or can a solution be proposed that is applicable to any genomic target? So the answer is in principle, yes. But if your method, for example, for some reason is not applicable to any of the 3 target families that the IDG is interested in, then that sort of a method is not going to be useful. So, for example, if your predictive method for some reason only works in protein transporters, in which case that is not going to be responsive. But in general, yes. I think in general also it might be useful to think about experimental validation in one of the 3 target classes. So that's not 100% required. But right. So it's important to remember one small change. So in the pilot phase of the IDG, we had 4 protein classes: nuclear receptors, kinases, GPCRs, and ion channels. In the current phase, the nuclear receptors are no longer part of the experimental setup. So the only things the IDG does experiments on are kinases, GPCRs, and ion channels. Okay, the next question is, if preliminary evidence is provided in the proposal of the ability of biocuration to shed light on previously uncharacterized biology of IDG targets, will biocuration of omics data be supported under this FOA? So I think the answer is yes, but in general, I would like you to think carefully about biocuration. So if your biocuration is so heavily dependent on human involvement, you have to think about what is going to happen after 2 years. So if it's a very dynamic field which changes very quickly in short periods of time, you need to be able to convince us and the reviewers that this biocuration effort done for the 2-year period is going to be a sustainable resource and be useful for the community in the long term. Okay, the next question. Can existing IDG members serve as collaborators or co-PIs in proposals? So this application is completely open. Nobody yeah, so it's completely open. I think one of our aims within the program is to expand the number of people, the number of groups, the different types of groups that are involved within the IDG effort. So it's an open thing, but we would also like to be inviting to the larger members and not be limited to the existing group. The next question is, what fraction of the proposal should be informatics tool development versus experimental follow-up studies? That sounds and looks like the first question, and I'm going to say it's the same answer. So let's see. I think there are a few more questions on the chat. If I can control the mouse. So it says, hi, thanks a lot for the overview. Is it required that visualization projects propose new data sets, or could visualization tools be designed to operate on the existing data? So I'm not sure what you mean by existing data. If you mean existing data that's available within Pharos, the answer is yes. If you mean existing data somewhere else, I don't know exactly how to interpret your question. But yes, you don't necessarily have to bring in new data sets. If you are happy and you have ways to visualize existing Pharos data, that's fine. But if you would like to bring in other data sets, that's fine too. Okay. Is there an expectation that non-IDG members reach out to current IDG members to get support or input on the proposed visualization tools to ensure that those new tools can be integrated? If your question is whether you need support letters from existing IDG investigators, the answer in general is no. You should use the Druggable Genome email address that was on the first or second slide, and NIH program staff would be happy to get you as much or as little or as much detail about what our intentions within the IDG program and within the KMC, the Knowledge Management Center effort, are, and how flexible Pharos can in principle be or in practice be now as well as in the future. So just as a reminder, Pharos is a developing entity, so it changes just like most other software engineering efforts change. We try to be current and open. We use as many open things as we can. And we are happy to, in general, work with people to get their things up on the website. Okay, so that's all the questions I see that is in the chat. I'm happy to hang around here for some more time if people want to ask questions. If you want to ask questions verbally, just unmute yourself. If you want to ask questions in the chat, you can do that too. Okay, somebody has one more question, but I don't see the question. Okay, so whoever is trying to ask one more question, we don't see the question. But if that was a mistake, then that's fine. Okay, so here's the question, I guess. Whoops. All right, that's a long question. So, is there an expectation that proposals include collaborators who are experts in drug development to come up with specific tasks of visualization, or can these be developed with members of the IDG after funding has been received? Okay. All right, there's another question. So, I'll make two comments. This is not a drug development enterprise. We are not going to be developing drugs. We will develop chemical reagents that's more chemical biology than drug development. They have to have certain characteristics. We will also be building biological reagents of various kinds. The aim is not necessarily pharmaceutical. It could be pharmaceutical, but primarily we suspect that the first thing you would need to do with understudied proteins is find out what they do, in what context, before you get into drug development. But beyond that, I think you don't have to have everybody already pre-identified in general. You can always consult later on. So I don't think, if it is not crucially dependent, that expertise is not crucially dependent on what you propose, you don't need to have them. Okay. And again, if my answers are not sufficient or you need more clarity, you can always ask it again or ask it later. So then another question comes, just to confirm that I don't necessarily need a supporting letter from an IDG member for this application, right? Yes, you do not need a supporting letter from an existing IDG member. It is not going to be helpful even if you include one. But, you know, I mean, I do want to emphasize, however, that, you know, you should read the links that we have provided. There's lots of data on what we are doing, what the experimentalists are doing. You should try to become familiar with it. The more familiar you are with what we are doing within the IDG program, the more responsive an application you can generate. But we don't need support letters. All right, so we might be close to a point of diminishing returns. So let me remind people that this whole webinar will be posted. We also have an FAQ of some kind. I think these questions that I have answered, we will put the answers up on the website as well. It should be there in a day or so. So you can see and point your friends and colleagues to these things if you want. There's another question. Go ahead. It's better to ask questions than to have silence. So, since the FOA is sponsored by NCI, does the experimental validation need to relate to cancer? So let me repeat this. This is a program that's funded by the Common Fund. NCI just happens to manage the RFA. Common Fund projects are relevant for all diseases and organs and whatever NIH is interested in. So no, you do not have to have your experimental validation be relevant for cancer, but that doesn't mean it can't be. It can be for anything you want. Yeah. Okay.
I have a quick question here. So if you have any future questions, about the webinar, should we contact you? I'm not sure if you saw your contact address or the presentation.
So there are two ways you can do this. The RFA lists Jean-Claude Zencluzen's contact. You can send it there, or the other one that we had, it's right here, help email druggablegenome@mail.nih.gov. So either one will get it to the right people. Okay, so I think if there aren't any more gone, you can always ask questions via email. I think we'll bring this to a close unless I see something in the chat in the next 5 seconds. It'll be like the shuttle takeoff thing. Okay, all right, so I think we'll bring this to a close. Thank you very much for participating. I look forward to reading interesting applications. Okay, bye-bye.
I was just going to mute it.
I probably screwed that up already.
Oh, it should. And just copy and paste everything. There we go. Copy everything. Oh, well, it's all recorded anyway, so you're good. All right. Sure.